Anir,
Yes.
we are having 5 more minutes. Let us wait for a few minutes. After that, we can start immediately.
Okay, okay, no problem. No
Yeah,
problem.
thank you. Thank you. Yeah. Anir,
Yes, Professor.
is it possible to change to presentation mode? It is now in slide mode.
Okay.
It is also showing the. Now it is okay. Yeah.
Yeah, but the problem is if I keep it here, then
And you can go to the present here. Here you can full screen mode. Yeah, you can go to the full screen mode. Let us see.
it's fine now.
Uh, Again, it's ஆமா. நம்ம கொடுக்கணுமா அவர் கொடுக்கணுமா? So go to the display setting which is appearing in the left top middle. Yeah, middle one, middle option.
Um,
That you can the zoom link you are seeing na mele in the top. Yeah, display setting.
this one. Oh, which one?
So everything is same as
Configure the settings for your display.
so otherwise what we can do. Yeah, first option you have to select swap presenter view and slide show.
This show the taskbar.
Yeah,
Or
yeah.
which-
Again go to the display setting. Display setting. Go to the display setting.
wait.
First
Yes.
option it is coming now. Swap presenter view and slide. Now it is fine. Yeah.
Okay, okay, okay. Thank you. I didn't know that.
Yeah. I also don't know that my scholar only helped me.
Okay, thanks to him or her. I don't know.
Yeah. So Anil, let us start.
Okay.
Yeah, very good afternoon to all. So let us now have the session 4 in the day 2, which is going to given by Anirban Chandra from Humboldt University, Germany. May I request Dr. Murugesh Pandian, Assistant Professor, Department of Chemistry, Bharatiyar University, to introduce the speaker. Thank you so much.
Thank you, Dr. Sundarvel, for giving opportunity. Giving me this opportunity. Good afternoon to all. I'm very happy to introduce. I'm welcoming you all for the second day, second session of FCS 2020. I welcome you all for the. On behalf of Department of Chemistry, I welcome you all. Our speaker is Dr. Anirban Chandra. Who obtained his MSc from Bordon University from West Bengal, India. And after that, he stayed 3 and 3 in IIT Kanpur for as a research fellow. Then he moved to for his PhD. He moved to Humboldt University, Berlin. He did his PhD under the supervision of Carol Drake. And he has published more number of papers, and particularly he published a high impact journal like Anglicum and JAX. Now I request Anirban Chandra to present his research findings. The title is temperature dependent oxidase reactivity, which is mediated by cobalt complexes supported on strain action core. Dr. Anirban Chandra, please.
Thank you, Professor. Thank you very much. Thank you for giving me this platform to share my research work with all of you. And good afternoon, everyone. So I am Anirban Chandra, and I am working in Humboldt University under Professor Ray as a PhD student. And today I am going to talk about some of my research work where we are I am specially involved in our group to development of oxygen reduction catalyst and with multinuclear metal complex system, and we use tanoxane core as a ligand backbone. And I hope you will find some interesting works in next few minutes. Okay, so at present, can you see something?
Yes.
Hello.
Yes.
Wait, but somehow.
Okay.
Excuse me, please. What happened? Okay, maybe here. Okay, so at present, humankind faces critical energy and environmental crisis due to the rapid depletion of the fossil fuel. And the environmental crisis originating from this fossil fuel crisis of the fossil fuel is actually fostering the research for the to search of the alternative fuel based energy, and therefore supply of the alternative energy source is very much important and is very essential to solve this environmental crisis problem. Mostly nowadays the source of energy is the burning of fossil fuel, and burning of fossil fuel create a lot of greenhouse gases, which is actually damaging the environment. And so the alternative and sustainable energy source and chemical process which are environmentally friendly is on high demand. And in this respect, the use of solar energy is also a very challenging task. And now lot of research groups, a number of research groups are working to this alternative fuel for the alternative fuel cell or solar based research. However, if we look into the nature, green plant can successfully convert and store the solar energy in the convert and store the solar energy via the photosynthesis in photosystem 2 using oxygen evolving complex like Mn4 manganese cluster, what Dr. Mondol already discussed in the morning via the Koch cycle, and it converting the solar energy by oxidizing water into oxygen. Whereas in cellular respiration, this oxygen is used for oxidation of glucose to producing carbon dioxide as water, and thereby producing also yielding energy. However, the in the S4 state of the Koch cycle is responsible for the formation of the O bond from water to produce the oxygen. So it is not only the formation water oxidation, but in the other way, O bond reduction to water, 4 electron reduction of oxygen to water is also important. It is not only in aerobic respiration but also in the fuel cell, because in fuel cell most of the in fuel cell the common cathodic reaction is the 4 electron reduction of oxygen to water. In any kind of fuel cell, mostly hydrogen based or alkali fuel cell, at the anode hydrogen is oxidized to proton, whereas at the cathode oxygen is reduced to water. However, the efficiency of the fuel cell depend on the high loading of the platinum metal in the cathode. The more loading of platinum metal is required for the higher reactivity of this fuel cell. And yeah, as we know, because of this, it cost the expense, the fuel cell is become more expensive because of the presence of this platinum metal. The another way of oxygen reduction is 2 electron reduction of oxygen to hydrogen peroxide, and it is also very significant because hydrogen peroxide is a alternative energy carrier, is can be used as an alternative energy carrier because of its high electron density at ambient temperature. It is also environmentally friendly oxidant and also important because of its delignification efficiency. But if we look the industrial production of hydrogen peroxide process via the anthraquinone method, it need palladium catalyst for the formation, for the effective formation of this catalytic cycle. And again, presence of the palladium catalyst increase the cost of the production. So we can see that the for the fuel cell for the generation of hydrogen peroxide right now, 4D and 5D metal are used, which always increase the cost, and also we have to compromise with the efficiency of these cells. However, if we look into the nature, we can see that several enzymes use cheap transition metal in their active sites, and they selectively oxidize oxygen to water or to hydrogen peroxide. Such as here, I show some example of cytochrome C oxidase, where iron and copper binuclear metal center is involved in its active side, and the trinuclear copper center is involved in lacage, which selectively reduce oxygen to water without producing of any partially reduced product, which is very important. And another copper based enzymes such as copper amine oxidase, galactose oxidase are responsible for selective 2 electron oxidase reduction of oxygen to hydrogen peroxide during the substrate oxidation process. Therefore, the cheap transition metals such as iron, cobalt, nickel, and copper has the potential to replace the expensive palladium and platinum metal for the fuel cell or for the hydrogen peroxide production process. So, for the several decades, scientists are trying to develop cheap, uh, cheap transition metal catalyst which can successfully able to reduce dioxygen to hydrogen peroxide or water. Here, I choose some of the selective example from the previously known literature, and you can see that the several groups are tried to develop the cheap transition metal catalyst, but still date there is no such efficient catalyst that can be used that can be used for an industrial purpose. So the research is still going on, and we are still in search of such more efficient catalyst for the future generation. And one thing I want to mention here, that all the catalysts reported so far in literature, almost all the catalysts can reduce oxygen in any one direction. So either it can able to reduce oxygen via 2 electron process to hydrogen peroxide or via 4 electron process to water. So if we can make one catalyst which can reduce the oxygen in both the way, depending on slight variation of the reaction condition, then it is also very much effective for the future generation. So nowadays multinuclear metal catalysts are in concern, Because multinuclear metal catalyst has higher catalytic efficiency, they can provide a closed structure where metal center can have extensive cooperative effect, and because of the synergistic interaction, multinuclear metal center are are much more effective compared to the mononuclear or dinuclear catalyst system. So, in my PhD, one of my research goal is to investigate how the what will be the role of the supporting ligand structure, and what will be the effect if we change the ligand structure of this core, and how it will affect the oxygen reduction ability. So in the next few slides, I will discuss the changing of the dendrimer ligand core structure, how the catalytic dioxygen reduction ability changes. For the multinuclear metal complex synthesis, we choose the stanoxane based stanoxane derivative of the ligand. So we synthesize two ligand system. One is containing the SN4O2 stanoxane core. Here you can see from the crystal structure 14 and 2 oxygen atom are involved and the core, and another one L2 which is containing the hexanuclear stanoxane core with 16 center and 6 oxygen center, and it from a drum shaped molecule. From the crystal structure of this molecule, it is evident that for the hexanuclear system, all these metal binding sites are equally distant from each other, and the distance is around 11 angstrom. Whereas for tetranuclear system, out of the four, only two center are close together and approximately 7 angstrom distance. The ligand, these two ligands are synthesized from this ligand precursor using the stanoxane derivatives. So after getting the ligand, I did the metallation with cobalt salt because I would like to work with because I work with the with the cobalt salts in my research work. So, with the reaction of cobalt triplet, this tetranuclear complex gives this tetranuclear cobalt complex, which is characterized by the tin NMR. Here in the tin NMR, we see for the complex, we see one doublet around minus 210 ppm and 213 ppm, which is exactly similar to the free ligand. So, this from the tin NMR, this is confirmed that the stanoxane core is remain intact after complexation. And from the EPR structure of the it shows a high spin S equal to 3 by half cobalt 2 center, and also from this EPR structure it is evident that the cobalt centers are not interacting between each other via any intramolecular interaction process. And then I do the oxygenation of this cobalt 2 complex of this tetranuclear cobalt complex, and upon oxygenation I found that there is one UVB absorption band start forming with absorption maximum 464 nanometer, and having the molar extinction coefficient value around 12,000 at minus 50 degree in acetone. The newly formed species is depends on the is reversible and is depend on temperature. So with increasing the temperature, the amount of yield decreasing, while with decreasing the temperature, the absorption spectra always increase. That means the species that is forming is also increasing the yield. The new species EPR silent, and to characterize more, I did the Raman experiment, resonance Raman experiment with 16O and labeled 18O level oxygen, and I found two isotopically sensitive vibration band at 862 nanometer and 862 centimeter inverse and 595 centimeter inverse, and 862 centimeter inverse band downshifted to 808 centimeter inverse upon 18O leveling, and also it's downshifted to 561 centimeter inverse upon 18O leveling. So from the 54 centimeter inverse downshift of this band, we correlate with the O vibration of a paroxysmal species, whereas 34 centimeter inverse downshift of the band we correlate with the metaloxo stretching vibration, and these two value exactly match with our DFT calculated parameter. To now to know more about this electronic and geometric structure of the peroxo species that we generated, we did the XAS measurement and along with the DFT studies. In the XAS measurement, this figure of cobalt K age gene spectra shows the blue line is for the tetranuclear cobalt complex, which is exactly matched with the reference cobalt 2 complex. So in the starting complex, cobalt is +2 oxidation state, whereas the newly generated peroxo species, which is showing here with the red line, is matched exactly with the reference cobalt 3 plus species. So that is confirmed that all the cobalt center in the tetranuclear system is oxidized to cobalt 3 upon addition of oxygen. And here you can see the slight increase in the pH shift for the peroxo species, which increase the more ligand field strength in the peroxo species compared to the tetranuclear complex. And in this figure, I show that the experimental and the simulated spectra for the exact simulation spectra of the tetranuclear cobalt species. The blue one and the red one is the intermediate. So from the exact parameter and the DFT calculation, we optimize the geometry of the tetranuclear cobalt complex, where cobalt center is coordinated with the 4 nitrogen and 2 triplets, so it's a 6 coordination. However, during when react with oxygen, it's become 5 coordinated species. As you can see that one of the nitrogen atom is not binding with the cobalt center, and it is the energy optimized model in the DFT. And in the peroxo species, the O bond distance is around 1.34 Armstrong. The same type of reaction was done with the hexanuclear ligand L2, so to form a hexanuclear cobalt complex. And this cobalt complex was also characterized by 11910 NMR, which shows a sharp singlet at minus 485 ppm, which is exactly similar to the free ligand NMR structure. So that also confirmed that the stanoxane core is remain intact after complexation. And EPR data also shows that high valent cobalt 2 signal with S equal to 3 by half spin state, and that is also that also confirm that the cobalt 2 centers are not interacting among each other, and there is no intramolecular interaction between the cobalt center. And the DFT optimized structure suggests again the 6 coordinated structure of the cobalt 2 with 4 nitrogen atom from the ligand and 2 triplet anion. And upon oxygenation of this cobalt 6 complex, we get a one UVV's absorption band at 470 nanometer. So the so from coming from tetranuclear to hexanuclear, the UVV's absorption band shift about 6 nanometer, but we see the molar extension coefficient value is around 28,000. Maybe it's because of the higher metal center is involved in this hexanuclear species. And this formation of this new species is also depends on the temperature, and it's a reversible process. The newly formed species is EPR silent. And EPR, to know more about the geometric and electronic structure, we did the Raman resonance Raman experiment and also the X-ray study. In the resonance Raman experiment, we get two isotopically sensitive vibration band at 868 centimeter inverse when we use 16 oxygen and 611 centimeter inverse. And these two band downshifted to 819 centimeter inverse and 584 centimeter inverse in presence of 18 oxygen. The 49 centimeter inverse downshift of this band is actually correspond to the O stretching vibration of the peroxo band, whereas 27 centimeter inverse shift is correspond to the metal oxo stretching vibration. And this data also fit very nicely with the DFT calculated parameters. From the cobalt K age genes data, we can see that the black one is for the hexanuclear cobalt complex, which is showing 1.6 electron volt blue shift when it's oxygen when for the peroxo complex. So 1.6 electron volt blue shift is confirming about the oxidation of the cobalt 2 center to cobalt 3 center. And you can here you can see that the pH value also shifted towards the higher electron volt for the peroxo complex compared to the starting complex, which also indicate that the higher ligand field strength in the peroxo complex compared to the starting cobalt 6 complex. From the DFT optimized except simulation and DFT optimized structure, we determine this energy optimized structure, and here the O bond distance is 1.39 Armstrong. Now, after successfully characterization of this tetranuclear and hexanuclear pieces and their intermediates, peroxo intermediate, we want to see how our complex behave towards oxygen reduction, whether it can able to reduce dioxygen, or at the same time whether it can be possible to use as a catalyst or not. And for this we use Fukuzumi and Guiller method. And so this method is introduced by Fukuzumi, where it is except of electrochemical method, you can check the oxygen reaction via UVBs by this method. So in this method we have to use uh, source of electron, where here I use the decamethyl ferrocene as the electron source, and proton source I use trifluoroacetic acid or triflic acid. And in presence of electron and proton, our my complex I set to react with the oxygen. And what I found is that I found when the reaction proceed, one new band start generating at 780 nanometer, which is corresponding to the decamethyl ferrocenium ion. And from the molar extinction coefficient value of the decamethyl ferrocenium, which is known, which is 520 mole inverse liter mole inverse at 780 nanometer, I did, calculate how much oxygen was reduced. But very interesting thing here is that I found out that the oxygen reduction is actually depending on temperature. So when I did the reaction, I did the reaction at variable temperature. When I did the reaction at low temperature, the amount of decamethyl ferrocenium formed is the highest. Whereas with increasing the temperature, amount of decamethyl ferrocenium form is gradually decreasing. And it is true for both tetranuclear complex and hexanuclear complex. And in this table, I shown that how much equivalent of oxygen reduced using 0.01 millimolar complex as catalyst. So at minus 50 degrees centigrade for tetranuclear complex, I found around 3.5 equivalent of oxygen reduced. So which correspond towards water, whereas at higher temperature, the amount of oxygen reduced is 1.92 equivalent. So 2 electron oxygen reduction take place, which is corresponding to hydrogen peroxide. Similarly, for the cobalt 6 complex at low temperature, you can see that amount of oxygen reduced is around 3.8 equivalent, which is also again indicate the 4 electron oxygen reduction, whereas at low temperature the amount of oxygen reduced is 1.8 equivalent, which indicate the 2 electron oxygen reduction to hydrogen peroxide. However, I did the proton NMR experiment to confirm how much hydrogen peroxide or water form in the reaction. So in this NMR spectra that I am showing, it is a control spectrum. So in the same reaction condition, in deuterated acetone in presence of acid decamethyl ferrocene, and in presence of my catalyst using oxygen, when if it's pure water in absence of any hydrogen peroxide, I got a signal at 4.60 ppm. And if when I increase the amount of hydrogen peroxide, then I see the signal is shifted from 4.6 to 4.4 to 4.0 to 3.9. That means with the amount of hydrogen peroxide, the signal is shifted. And then I test the reaction mixture after the catalysis for tetranuclear cobalt complex at minus 50 degrees centigrade. I saw the signal at 4.20 ppm, and this 4.20 is in between the 4.04, which is for 30% hydrogen peroxide, and 4.44, which is around the 15% hydrogen peroxide. So from here, I conclude that around 20% of hydrogen peroxide form for the tetranuclear cobalt complex at minus 50 degrees centigrade. Whereas if I increase the temperature and when I did the catalysis at 30 degrees centigrade and I measured the NMR, I found the signal is shifted to 3.8 ppm. And for the 70% authentic hydrogen peroxide at the reaction conditions, the signal come at 3.9 ppm. And so I conclude that more than 70% of hydrogen peroxide, around 74% hydrogen peroxide is formed here. For the hexanuclear catalyst at minus 50 degrees centigrade after the reaction, potential data shows a signal at 4.68 ppm, which is exactly matched with the authentic water signal. So here I conclude that for hexanuclear cobalt catalyst at minus 50 degrees centigrade, no hydrogen peroxide form, and it is a 4 electron complete conversion of oxygen to water. Whereas at 25 degrees centigrade, the signal is 3.92 ppm, which is exactly matched with the 70% hydrogen peroxide. So from this UVV's experiment and the protonenema study, I came to the conclusion that this catalyst is able to reduce oxygen depending on the temperature. At low temperature, it's favored the 4 electron reduction pathway, as you can see from this from the equivalent of oxygen reduction amount, and at high temperature, it prefers the 2 electron reduction pathway. And and it is the cobalt 3 peroxo species that is involved in both the cases is the control the this 2 electron versus 4 electron reduction pathway. The stability of the cobalt peroxo species is the controlling factor for this 2 electron versus 4 electron reduction pathway. And to prove that, I did some proton transfer and proton couple electron transfer experiment. So what I did at room temperature, with this intermediate, I put some decamethyl ferrocene, I add electron, and I see that there is no decay of this peroxo band. That means at room temperature it doesn't react with any electron. However, if I put some acid like trifluoroacetic acid, I saw immediate decay of the peroxo species. As you can see from the spectra, at 25 degrees centigrade, when I add acid, the characteristic band of 464 nanometer for the tetranuclear species and 470 nanometer for the hexanuclear species start decaying. And this decay follow a nice first order kinetics, and from this value I can able to determine the second order rate constant. Whereas when I check this same reaction at low temperature, I found that this peroxo species is stable in presence of acid if there is no electron, source in presence of only electron if there is no acid. So as you can see, if I add acid, there is no reaction. If I add only electron, there is no reaction. But if I if I add both the proton and electron together, then the decay of the peroxo species take place. So that is suggesting that at 25 degrees centigrade, it goes via proton transfer process where this metal oxo bond cleavage take place to produce hydrogen peroxide. Whereas at low temperature, in presence of both proton and electron, proton couple electron transfer takes place where O bond cleavage occurs to form the water. And I determine some more uh, proton transfer rate and proton couple electron transfer rate at different temperatures. So for the tetranuclear complex, at low temperature, you can see PCT is the predominant factor, as I showed here. Whereas at high temperature, proton transfer is the operate very drastically. And the transition temperature I did experimentally observed is 11 degrees centigrade. Whereas for hexanuclear cobalt complex, the transition temperature between proton couple electron transfer and proton transfer process is around 20 degrees centigrade. However, when I determined the turnover number, I found that hexanuclear cobalt complex is more efficient at both minus 50 degree and 25 degree centigrade with turnover value 65 and 16, whereas the for tetranuclear cobalt complex, the turnover number is not that much that to the hexanuclear species. So from this experimental observation, I propose this catalytic cycle where cobalt 2 species first react with oxygen to form a dinuclear metal peroxide species. Now this dinuclear metal peroxide species at high temperature goes via proton transfer process in presence of proton to produce hydrogen peroxide, and electron is required to reduce the cobalt to cobalt 2. Whereas at low temperature, at minus 50 degree centigrade, it goes via proton coupled electron transfer process to produce water. And for hexanuclear species, we didn't see any kind of hydrogen peroxide at low temperature, whereas at minus 50 degree centigrade, tetranuclear species show around 20% of hydrogen peroxide. So in this study, I showed that if we change the ligand backbone, then there will be a drastic change in the catalytic efficiency of the oxygen reduction reaction. So far I showed you the tetranuclear ligand, which is containing the SN4O2 backbone, and hexanuclear ligand, which is containing SN6O6 backbone. And from these two ligand, I synthesized two cobalt complex, and after oxygenation, I get one endon peroxo complex, and here the endon peroxo complex for the hexanuclear complex is 2. So we are also interested to see how it's reactive towards the external substrate. So since it's a peroxo species, so immediately I try the nucleophilic oxidation reaction, because peroxo species are well known to show nucleophilic oxidation reaction. And this is the reaction with tetranuclear peroxo species with benzaldehyde, and you can see the decay of the characteristic absorption band, which follow a first order kinetics. And from this k observed value, I determine the k2 value, second order rate constant k2 for benzaldehyde. The same experiment were performed using different para substituted benzaldehyde, and from the different k2 value, I determine the Hammett plot. And you can see the Hammett plot is positive, uh, which confirm that its nucleophilic behavior. The same kind of experiment was also performed with hexanuclear peroxo species, endon peroxo species 2, where the decay of 470 nanometer band was observed, and from the different K2 value when we plotted, we found the positive Hammett constant value, which confirming about its nucleophilic behavior. Now, interestingly, I check some electrophilic oxidation reaction with these substrates. Generally, metal peroxide species are not known to show any kind of electrophilic behavior. Very few examples are only known in literature where any kind of metal peroxide is showing electrophilic behavior. Uh, but in nature, metal peroxide species are known to show the electrophilic behavior. So when I test my complexes with this substrate, I show that my complexes are react with the electrophilic substrate. These two intermediates act as an electrophilic oxidant towards this different substrate. However, you can see from this table that the peroxo complex 1 is not able to react with the stronger CH bond such as fluorine, where bond dissociation is around 81.2 kilocalorie per mole. However, the peroxo complex 2 can react with the stronger CH bond. So it is confirmed that the endon peroxo species is a strong oxidant compared to the peroxo complex 1. To get further insight, I determine the KI value using xanthine and D2 xanthine, and KI value is around 2.3 for the peroxo species 1 and 3.02 for the peroxo species 2. The positive KI value confirm that the CH bond abstractions take place at the rate determining step of this reaction. Now, there is a question that can arise whether it is a peroxo species that can swing the electrophilic oxidation reaction, or the peroxo species is partially converted to the metal high valent metal oxo species, which is responsible for the electrophilic oxidation reaction. To check this possibility, I did the oxo transfer reactivity and also CH bond activation reactivity in presence of 18O level water. It is well known that this metal high valent metal oxo can exchange its oxygen atom with the 18O level water, and if any high valent metal oxo species is involved in the reaction, then I will get the product with labeled oxygen. But the GC analysis doesn't indicate any kind of 18 labeled product. That so I confirm that it is the metal peroxide species which is showing the electrophilic reactivity. Also, another thing is that if we look into the literature, we can see that for the metal oxo species, the Ki value are relatively very high. This is the example from the Professor Nam group, where cobalt 4, where they reported cobalt 4 oxo in presence of scandium, and there they observed the Ki value for the around 13, but here you can see the ki value is relatively smaller. So this low ki value and also the 18O water experiment confirm the fact that there is no high valent metal oxo species involved in electrophilic oxidation reaction, and it is the only end on dinuclear cobalt peroxo which is showing this electrophilic reactivity. And now I would like to show some phenol oxidation reactivity with the intermediate 2. I didn't choose the intermediate 1 because it is not as much strong electrophilic oxidant as 2. So all the phenol oxidation reactivity was done with this only complex 2. And I choose a series of phenol for the oxidation reactivity. In here I show the reaction of this endon peroxo species 2 with 2,6 dideoxyributyl phenol, and you can see the addition of excess amount of phenol decrease the characteristic absorption band, which is follow a first order kinetics, and from there I can determine the second order rate constant value K2. Now, to know whether this wedge bond activation take place at the rate determining step or not, I determine the Ki value using using phenol and its deuterated version, and also 4-methoxy 2,6 dideoxyributyl phenol and its deuterated version. And we see that the Ki value is almost similar, that we around 2.93 and 3.09, and that we already such low Ki value we already observed during the electrophilic oxidation reaction. So to prove again whether there is formation of any high valent metal oxo species or not, I did the reaction in presence of 18O level water, and I didn't see any kind of 18O incorporation product in GC. And so from the lower Ki value and 18O level experiment, I conclude that there is no high valent metal oxo species forming in the reaction, and it is the only dinuclear cobalt 3 peroxo species which is responsible for the phenolic wedge bond activation process. Now, um, if we want to explore the mechanism how the phenol oxidations take place, so there are three possibilities for the transfer of proton from neutral phenol to the endon peroxo species, either via electron transfer followed by proton transfer followed by electron transfer, and concerted hydrogen atom transfer or proton coupled electron transfer process. So in the literature, Bronsted-Tafel analogy shows how to determine which way the proton transfer takes place. So with the when so for if we plot RT by F ln K2 versus E, if it come around minus 0.5, then it's a pure electron transfer. For if this value come at minus 0.1 for a pure proton transfer process, but for PCET process this value should lie in between minus 0.5 to 0.1, but for hydrogen atom transfer process this value should be constant. So I also take different phenol and I react with this peroxo intermediate, and what I found is that plot of T by F ln K2 versus Yox shows a nice linear correlation, where I found that the slope is minus 0.285. So you can see that there is no evidence of this kind of slope here. However, there is a paper from Professor Carlin and also Fukuzumi, where they already mentioned that such lower value of the slope can be possible, and this is happen um, when the proton transfer from neutral phenol to endon peroxo species take place via the concerted electron transfer proton transfer pathway. But here there is no complete transfer of charge taking place, it occurs via the partial transfer of charge. So here I conclude also that for my endon peroxo intermediate 2, the phenol oxidation is take place via the partial transfer of charge. N- uh, Okay, now to find out the more details about the mechanism, I determined the did the product analysis with normal oxygen and also 18O level oxygen, and I found that in almost every case 1,4-benzoquinone is the major product, and for 2,6 ditertiary butyl phenol and 2,4,6 tritertiary butyl phenol, we also found this dibenzoquinone product. For the reaction with 2,4,6 tritertiary butyl phenol, at the end of the reaction, I also get EPS signal with G value 2, which indicate the presence of a free radical of the 2,4,6 tertiary butyl radical system. So from this observation, I propose the mechanism where this phenol first react with endon cobalt peroxo species to form a phenoxyl radical, which I get evident from here, and which this phenoxyl radical can dimerize for some cases to form this product, whereas this phenol radical can again react with the endon peroxo species to form 1,4-benzodiquinone product. However, for the formation of the 1,4-benzodiquinone, there is two possible mechanism. If we use some kind of methoxy group with oxygen, then we can see the formation of the benzaldehyde, and if we use some alkyl groups such as tertiary methyl, then isobutylene should release. But since the reaction is stoichiometric and amount of isobutylene form is really less, that I can't detect it in GC. However, I am successfully able to detect the formaldehyde in the UV experiment via the NAS experiment. So here, just I would like to explain this black spectra is for the starting cobalt complex, whereas this red one is for the cobalt peroxo species. And when the reaction was done with the 4-methoxy 2,6 ditertiary butyl phenol, this spectra decay to this gray color spectra, and then into this solution, when I add the NAS reagent, I can found a band is generating with increasing the temperature at 413 nanometer, which confirm the presence of formaldehyde. So from the product analysis, from EPS signal, and from the detection of formaldehyde, I I predict that this mechanism is quite valid for this phenol oxidation by the endon peroxo species 2. So now I would like to finally conclude my talk, where I showed you that how the ligand architecture change of ligand architecture can affect the dioxygen reduction reaction, and it's not only affecting the dioxygen reduction reaction, it also influencing the reactivity towards electrophilicity and nucleophilicity. When we have the more constraint, so for hexanuclear complex, the distance between the ligands are 11 angstrom, whereas in tetranuclear complex the distance are around 7 angstrom. So for the peroxo species formed from the hexanuclear complex are suffer from more constant, and also because all these center are equidistant, so maybe there is a more synergistic interaction or cooperative effects that is operating, which is the key for its better electrophilicity, better electrophilic character. And finally, I would like to acknowledge my supervisor, Professor Ray, and my group, and the financial support from Unicat, Unisyscat, and also the Humboldt University. And thank you for all your kind attention.
Thank you, Anirban, for nice presentation. Any questions from audience? Anirban, I have one small question. Why you are getting in tetranuclear two tin peak, and whereas for hexanuclear you got a one single peak in tin NMR?
Yes, just one moment. Yeah, so for the tetranuclear system, if you look, this there are two types of tin center present. So these two
Okay.
tin center are connected with the two oxygen atom, whereas these two tin center are connected with the one oxygen atom. So there is not a huge chemical shift, but the signal is just pleated.
That is a oxo bridge or hydroxo bridge?
No, this is oxo bridge.
Oxo bridge. Okay. And in this cobalt complex, the cobalt is a plus two oxygen state, no?
Yes, cobalt are
It's
plus
a
two oxygen.
paramagnetic or diamagnetic?
No, it's a
Paramagnetic.
paramagnetic. That's why it's CPR active also.
Then you are getting tin NMR also, no?
Yeah, tin NMR is also, there is no problem with tin NMR, but I didn't quantify the proton NMR because its proton NMR is a huge shift for the paramagnetic species, but tin
Okay.
NMR has no problem.
So when you add a cobalt, it doesn't disturb the stenoxin core.
No, that is the reason I did the NMR with the free ligand and with the complex, and I saw that that NMR is same. There is no change in the NMR, and that is confirming that the stenoxin core is remain intact before the reaction and after the reaction.
Reaction. Okay. Okay. Have you got any single crystal for this cobalt attached stenoxone?
Oh no, I didn't. We tried a lot, but it didn't work finally.
Okay. Thank you.
I am having, I need some clarification.
Yes,
Hello.
please. Yes,
Yeah.
Professor, please.
Yeah. So my first question is, so what makes the cobalt peroxide electrophilic towards electrophilic?
Okay, so maybe I can show you with my hand little bit. I cannot explain. So the thing is, what happened in biology, um, there are maybe lot of protein involved, which is we don't know why in biology it's showing electrophilicity, but in if you look into the literature, there is only one peroxo species from the professor Srivastav of IIT Guwahati of manganese peroxide. He reported that there is the electrophilic reactivity. But here what happened, so we have a stanoxane core like this, where you can see these two are cobalt center, and for the hexanuclear or tetranuclear, so these are like 7 angstrom or 11 angstrom apart. And when I add the oxygen, this coming close, very close to each other. So there is a strain. The stanoxane core is same, but you can see there is a strain. And maybe because of this strain, this peroxo species act as a electrophile.
Only that is the reason, or is there any role of first upon CH bond activation, the proton is transferred to the peroxo and making it as a hydroperoxo, and the hydroperoxo is electrophilic, or so on?
Um, but how to check that one? I mean, we only I only check that if there is any possibility for high valent metal oxo or not, because otherwise this hydroperoxide, that also thing, but I don't know. I mean, I didn't check that thing whether it's possible, because in presence of when I add excess amount of proton in the cobalt peroxide, as I showed you, at this peroxo band is completely decay. I mean, during this oxygen reduction reactivity. So if it have to form hydroperoxide, then it should be stable at some point. Otherwise, if proton is coming and bind with this form kind of hydroperoxide, something like this, then it can completely break the peroxoband and it's drastically decay. And I don't think then it will react with increasing the amount of substrate. It will give a linear correlation of the K2 value.
Okay. So suppose if you are equilibrating the cobalt stanaxene core prior to O2 reaction, what with the proton, what would happen? Whether it will form the same.
Can you
Suppose if
please?
you are taking the cobalt stanaxene core and you are equilibrating with the molecular oxygen and you are ending up with the peroxide intermediate. Suppose you are pre-equilibrating the complex with the proton, then you are allowing the O2 to react with the proton. In presence of proton, the stanaxene core,
Yes.
whether it will give the same.
Yeah. That reaction we did during this was one question from the reviewer, and we didn't see any kind of change. So it was in presence of proton. So first we take the cobalt solution and then we add proton, triflic acid or perchloric acid, and then we add oxygen, and we found that the equal amount of formation of the product. And then at low temperature, but at room temperature it's always there is a self decay involved, so there is nothing. So when we did at low temperature, we see that the formation of product is same, and from there we conclude, we give the answer that this proton is not making any difference for the formation of this peroxo species.
Okay. A few more questions are there. Now,
Please.
Bala, you can unmute your mic and you can ask the question directly. Bala Murugan?
Okay, I have. May I ask?
Yeah, you can ask Doctor now. Yeah,
okay,
sure.
Okay. So, Anirban, so when you add ferrocene, so yes, so that time your tin in stenic that does not get reduced to stenous.
No, Professor. Professor, here I would like to say that my complex is not able to reduce ferrocene. So ferrocene has the stronger oxidation potential. So we use this all the three ferrocene derivative, ferrocene, dimethyl ferrocene.
Yeah, means I am using when you use the decamethyl ferrocene. That also
yes,
that does not reduce.
No. That doesn't reduce. That doesn't reduce the stenoxane core. That doesn't reduce because I didn't wait. I can show you this spectra once more. Yes, so here this spectra I didn't show this you can see this 470 nanometer band is almost same. There is no change in this band. If I show this one, then you can see. So it's only little bit decreased because of this dilution effect, but there is no change in the stenoxane core. I mean, UVB, there is no change in UVB spectra. That's why we conclude that there is no change in the stenoxane core.
Okay, and when you plotted that K2, so when you that particular plot, so why you see a intercept?
Which one? Can you please tell
It's
me?
like in this case, like yeah. So next type, like in this case, so when you have plotted the rate of reactions with the substrate concentrations, so why do you see the intercept? Why it is not passing through the origin?
Yeah, exactly. So at 25 degrees centigrade, my complex suffer. I mean, these peroxo intermediates suffer from the self decay. So so I have to consider the self decay rate, and this is the self decay rate. So
Okay, so
here
that means if you subtract the self decay rate, so it will pass through the origin.
Yes, exactly.
Okay.
Exactly.
And so one more question. So in your NMR, when you quantify water and H2O2 by NMR, so but your solvent also will contain some residual water, right?
We I try
Like
my
any
best.
deuterium solvent, whatever we buy, that also contains some sorts of residual water.
Yes,
So
but
means have you?
yeah, that I did a lot of this blank and all these thing, and after buying this solvent, we always distill the solvent, and then after purging argon. So I think I try to minimize it as much as possible. But yeah, you are right. It
If
can
you
be
do a
possible.
water suppression, if you do an water suppression or water gate experiment, so can you do without drying your solvent? Means that also As you know, the deuterium solvents are very expensive. So if I further process it from a commercial source, we will lose our lot of quantity. So
Yes.
means I mean to say, if you do an Watergate experiment, so can you avoid that residual solvent which is present in your NMR solvent?
Oh, this.
The water suppression we can do by our NMR spec instrument.
Oh, okay.
So
Maybe
by that way, can we do it? Means I don't know. I'm just curious to know.
yeah. I am writing this thing. I will ask maybe water
And
suppression.
one more thing, that H2O2 you have quantified means through NMR, but can you quantify by iodide
Yes,
test?
yes. That I
Iodide
didn't show,
hydration.
but yeah. That we did. That we did. It gives a peak around 300 something 13 or
53
something. I
something
think.
like that.
Yes. In
Yeah.
presence of sodium iodide, in presence of excess proton. Yeah, that we did. That I didn't show here, but in our both paper is this is the this spectra are there. We confirm with formation of triiodide. We see the formation of triiodide.
Mm-hmm. Mm-hmm.
Yes, it is there.
And your last part of your talk where you have seen the electron transfer, yes, sir. Yeah, here. Probably you got a slope of minus 0.28, which is
Yeah,
lesser than an electron
yes.
transfer.
Exactly. This one.
So this is an asymptomenous electron transfer, right? Means what do I mean by that? Maybe it's not a pure electron transfer, it's not a
Yes,
pure proton transfer.
ex-
So maybe a 60% electron transfer and then 40% proton transfer.
yes. So I am still reading on literature to get more idea about it. But that is, I think, that is the thing you are telling about this partial transfer of charge. So
Yeah, partial transfer of charge.
yes.
So it's now become a very interesting part in this particular area of research. So sometimes it
Okay,
may not be a pure CPT or TCT or hat reaction. So,
okay.
so means if you consider those parts, then maybe you will get a good fit uh, with all other substrates. Means this type of research, I know that it is going on. Is one of my junior. He was working with Professor Andy Horowitz's labs, and he was working on the same type of project where
Okay.
he has proposed that type of thing. Means it is not yet published, but I think they have submitted the paper in some journals.
Oh, okay. I actually this one I only get from this Carlin paper where they
Mhm.
showed this copper superoxo phenol oxidation and there only one line. They also get the slope around minus 0.29.
Yeah,
So
yeah,
and then
yeah.
from there I just get the idea that okay, partial transfer of charge is possible. But truly speaking, I am still in search of more knowledge about how this partial transfer thing happen and all this. Mean I really don't have much idea
Mhm.
about this partial transfer of charge. So I just took the note from there and I put it here.
Okay. Thank you.
Yeah, yes, thank you, sir.
Uh, Yamuna, can you please read the questions?
Sir, our participants have few questions, sir.
I am not sir. First of all, please call me through my name and ask me whatever you want.
Uh, have you tried to use water or any other proton donor rather than ferrocene?
No, ferrocene as act as a electron donor. So for proton donor, I only use acid, and I also did the experiment with different acid, like very strong acid, like triflic acid, weak acid, like tetrafluoroboric acid, and both the acid different pK values, and I didn't see any change in the amount of product. So only the rate is little bit slow or faster, but the final amount of product is always same, no matter whatever acid we use.
Okay, sir. And the final question is, have you ever tried 2-electron water oxidation to H2O2?
Water, my complex is not showing any kind of water oxidation. It's showing oxygen reduction.
Okay, actually, the participant have asked this.
Yeah, no. Yeah, just I want to say, just one moment. Yeah. So in this reaction, you can see this two electron reduction of oxygen take place to hydrogen peroxide, and that is with
Actually, he
temp-
mentioned that it is just an hypothetical question.
Oh, okay. Yeah. But I don't check my complex in water oxidation. Yeah, if it will be very nice to check it whether or it this complex can show some water oxidation or not. But yeah, so far I didn't do anything. Yeah, and my group is also not working in this field, so yeah, I don't have any chance to check it.
Okay, thank you, sir.
Nirvan, I have small. Actually, when you use strong acid, it won't disturb the transaction core.
No, Professor. Of course, I didn't check the NMR after addition of the acid
Yeah.
because this cobalt is getting hydrolyzed or cobalt 3 hydrox or something. So I have no clue.
Mm-hmm. Okay.
But we only see that there is after addition of acid or there is no change in the UVB spectra.
Oh,
So
then it should be stable one.
yeah,
It should be.
that's the reason we believe and we also report that it's stable in presence of any acid or also in presence of base. I think I also tried that experiment too.
Okay. Thank you, Anirban, for a very nice talk, and
Thank you,
really, it's we enjoyed a lot. Okay, and
thank you.
on behalf of FCS, okay, FCS 2020 team, I like to thank you for your presentation and other people's for attending this webinar. Okay, we can gather after 5 minutes for next session. Okay, thank you.
Yes, Professor.
Thank you.
Thank you,
Thank you.
thank you, Professor. Thanks to all.
Thank you so much, Anis.
Thanks.
Yeah, I will call you later.
Yeah, sure. Thank you. So I can leave the meeting.
You can join the meeting for the next session. There is
Yeah,
no
but
problem.
now I can leave.
Yeah, you can quit your presentation.
Yeah, thank you.
Yeah, thank you so much. Gopal. Gopal.
Yeah, சொல்லு கேக்குது. Yes. Hello.
Just share your slides.
Okay. So I need to share.
Can you hear me?
Screen. Yeah, I can hear you. Can you? So you can see my screen?
So wait a minute. Yeah, it's yeah. Just open your presentation. Then only. Yeah.
Is that fine?
Yeah, it's fine.
Okay. So
Just for a few minutes after that, we will resume the conference.
yeah, sure. Okay. I also started my video. Is this clear? No. It's not clear. Okay.
Yeah, everything is clear. There is no issues.
Yeah, okay. So I can close my videos when I am presenting?
Yeah, you can.
Okay.
There is no issues.
Yeah.
The slides are very clear, so there is no need to.
Okay, then it's fine.
So very good evening to all. Now we are resuming for the session 3 of the day 2. So today this last session is going to delivered by Dr. K. Gopal Swami from RMIT, Australia. Now Dr. Murgesa Pandian will introduce the speaker.
Thank you, Dr. Sundarel, for introduction. Okay. Good evening to all. Welcome you all for the second session of this afternoon. Our speaker is Dr. K. Gopalsamy. Dr. K. Gopalsamy. He did BSc, MSc, and MPhil from Madurai Kamaraj University. Then he obtained his PhD from CSR CLRI Chennai under the supervision of Dr. V. Subramanian. Then he moved to USA, University of North Dakota, for his postdoc for 1 year. Then he did postdoc in ISET Tripadi for 1.5 years. Then currently he moved to currently he's working as a research assistant from October 2019 in RMIT University, Melbourne, Australia. He has published a lot of papers in well-known journals like Journal of Physical Chemistry and Journal of Chemical Physics, etc. And he received its DST National Postdoctoral Research Fellowship. And his research interest is computational material science. Now I request Dr. K. Gopalsamy to give a presentation.
Yeah, thank you, Doctor, for the kind introduction. And good afternoon to everyone. Today I'm going to talk about how metal organic framework material used to store separate gas molecule from computational point of view. So this is Melbourne City. It's an aerial shot, long view. It's a clear view of Melbourne City. So it's one of the beautiful city in Australia, and it has a very unique, famous railway station, and our university is located here. You can able to see my cursor.
No, yeah.
Wait, I'll take spot. Okay. So is this clear now? Can you hear me? Hello?
Yeah, you can proceed.
Yeah, okay, thank you. So our university is located here. Yeah, it's a overall view of the city. And I would like to share something about other universities located in Melbourne. The university, other than RMIT, Monash is there, University of Melbourne is there, Deakin, and a lot of all these universities are famous in Melbourne, and they are offering very good professional as well as diploma courses. Those who are really want to pursue a diploma and as well as professional MS and PhD degree, you can apply in any one of these universities. I think the admission is going on now. Apart from IELTS, PT is there. You can check in their website. You don't. I think you don't need to have like 88 plus or 80 plus for the PhD admission. It will be like 60 or 65 plus. So you can get a very good working environment in Melbourne, all the Melbourne universities. So if you are interested, please have a look. Okay, so with this, I would like to move to the topic. And yeah, so here in this slide, I would like to show the advantage and growth of computational chemistry as well as the supercomputer. So once upon a time, this one is supercomputer for us. So if someone is having computer like more than 3 decades, it is super, and for them that was supercomputer. So in order to use this old version of supercomputer to do computational calculation, we need to spend more than a month to finish a single calculation. But after now, the situation is totally different. The meaning of supercomputer is really got the real meaning. So here you can see the clusters of computers, hard drives, and by using this, we can do various computational calculation. If we want to do calculation with more than 1,000 atoms, you can finish the calculation within a day or within a week, depending on what we want to do with the calculation. So in the previous slides, I showed you the computer, what do you call, supercomputer hardware. So this is the component present in a single cluster. We call it as a cluster. The cluster is a combination of nodes. Node has multiple cores. So and it has a huge number of high number of RAM. So in order to perform calculation super fast, we need to have at least 4 to 10 nodes with multiple cores. Then only we can able to run the calculation and finish the calculation within a time period. So all these nodes connected with one master, they called as a master. The master will direct node how to run the calculation. So the master actually do the job. When we are submitting jobs through our computer, that will go to the master. That master will separate and determine how to run the calculation. The calculation will be run in all the nodes, and in the final, all the output sent to the master. The master cumulatively collect all the data and it will return to us, and we can monitor the progress of the job, whether it is finished or not, by using our computer. So in order to do computational chemistry or computational material science research, we need a laptop or computer. Even by using cell phone, you can monitor your status of your running job. You need a very good Wi-Fi and IP address. So that is more than enough. You can submit calculation from anywhere in the world, and you can access your data, and you can analyze your result. You don't need to go to your lab actually. So you can do whatever you want to do from science, from your home or wherever you are from the world. So in order to do this, use this supercomputer, we need software. So for that, we need a Linux platform, because all these supercomputer are installed with the Linux software. So we need to communicate or connect the supercomputer by using a Linux platform. So we need to give commands. By using that commands, the job will run. You can submit job, you can download the data, or if something went wrong, you can delete by using Linux commands. So in order to do computational research, you need to have basic computational Linux keywords. It's more than enough to handling supercomputer. So here you can see a list of softwares. So here I would say this CP2K, LAMPS, and Siesta, Quantum Espresso, VASP. So all these softwares are unique, and they are mostly used for material science. Like if you want to calculate the band structure or electronic magnetic properties of any narrow material, we can use these softwares. VASP is licensed version. That means we need to pay for the to get the software. But Siesta, Quantum Espresso are free software. We don't need to spend any money. We just download the software and we install it in the supercomputer, and straight away we run our calculation. And this one, GROMACS, it's for if you want to calculate study the properties of biological system. What do you call, if you want to calculate the protein-protein interaction, excuse me. or protein nanomaterial interaction, you can use GROMACS. It's a free software. And if you really want to do quantum chemical calculation like DFT, we need Gaussian. You can use Gaussian and DMOL3 and this one, ARCA. So Gaussian is a paid version, Gaussian also a paid version, but ARCA and DMOL3 also paid version. It's implemented in Material Studio. And this one, ARCA, Gamma US are a free version. We can download it, and you can straight away use the software. And yeah. Okay, so I'm slowly going into the topic. So before talk about metal organic framework, it would be nice we can talk a little bit about the conventional as well as well-known porous material. We all know about activated carbon, zeolite, and clathrate hydrates. These are all comes under porous material category. So these activated carbon widely used to remove dye from the solution or metal ion, and zeolites are widely used in industry for catalysis purpose as well as separation purpose. Clathrate hydrate also used for molecular storage as well as separation. Though these porous material having wide application, they have their own disadvantage. For example, activated carbon, it has a variety of pores like micro, meso, and macro pores, but it doesn't have any proper pore. That means it don't have any unique pores. And zeolite, you can't tune or tailor the pore size of the zeolite, and it has practical issues to use zeolite for all the application. Clathrate hydrate also, it's like a water framework, and you can't use it for higher temperature. So we want to store or we want to separate any gas mixtures. That material should be like more stable as well as user friendly. So I will talk that in the upcoming slides. So with this brief introduction, I'm just moving to metal organic framework. Okay, metal organic framework. This field got established after around 2000. Yeah, it started from 2000. The first metal organic framework synthesized is MOF5. It was synthesized by Professor Umar Yagi. In their group, they synthesized numerous number of metal organic frameworks with various topology. So it has two parts in the framework. One is connector and another one is linker. Here, connector is always a metal or metal oxide or metal clusters. Linker is it will be it is organic acid. It may be a dicarboxylic, tri, tetra, or polycarboxylic acids. And some cases they may have a azo group, like metal organic framework, covalent organic framework also there, zeolitic imidazolite framework also there. So today I'm going to talk about only metal organic framework. Here you can see crystal structure of MOF with various topology. So we can synthesize or we can produce various MOFs by choosing appropriate linker as well as connector. Yeah. Also, it's also depending on the synthetic procedure. Most of the MOF material synthesized by using solvothermal analysis method. The main advantages of these metal organic frameworks are, it's more interested because you can tune the property by changing the metal in the connector or carboxylic linker, or you can do functionalization on the linker in order to use this metal organic framework for specific application. Also, these metal organic frameworks are highly stable. That means it's they are stable up to some metal organics are stable up to 600 degrees Celsius. So most of the metal organic frameworks are stable up to 400 degrees Celsius. And main one of the main advantages of having this metal organic framework is its surface area is too high. For example, if 1 gram of this metal organic framework, if we have the surface area is equal to a football ground size. So this will highly helpful for the adsorption or separation studies. So before going to further, I would like to show something how this crystal structure looks like. I have shown two-dimensional structure. So here I would like to show you three-dimensional structure. So here is a two-dimensional. If you are simply rotating, you can see the three-dimensional structure of the MOF. It's a MOF5 structure. So it has unique pore, and the gray color are metal. You can see all the sides, and I would like to show you the supercell. That one is unit cell. This is supercell. So these metal organic framework are extended crystal structure. So it extended in the 3 direction, X, Y, and Z direction to the infinite distance. So, and this is for MOF5, and this one is copper BTC. Here connector is copper pedal wheel type connector, which connected to benzene tricarboxylic acid, and it's forming this beautiful structure. And this one is zirconium oxide. So these zirconium based MOFs are highly stable one, and it's highly useful to do studies near room temperature or next to room temperature. If you want to do some sort of study, we can use this MOF. These MOFs have its own application as well. So yeah. So these are the advantages of using MOF. And one more thing here is how computational chemistry play an important role for these MOF studies. In order to synthesize any of these MOFs, now the MOF synthesis are well established, and if really you want to synthesize MOF for particular application, yes, you can do synthesize, because there are plenty of MOF, more than 10,000, 15,000 MOF crystal structures are reported in the Cambridge crystal database. So it is it's all now open, like it's like open, whatever you want to do you can do. But how computational chemistry play a major role here is if you want to do any modification, for example, if you want to do functionalization over here with OH or any methyl or ethyl or anything, you need to go through with all the synthetic procedure that you need to synthesize. You need to have the connector linker separately. You need to synthesize the MOF. Then you need to do the functionalization. It will take much time. But as a computational chemist, I can just select the molecule and I can do the modification here by using appropriate software. I will do optimization. Then I will get the crystal parameter ABC values. Then I will go for the further study. Like if I want to do the electronic structure or band structure calculation, I can do. If I want to do the gas storage, I can do. So I can compare the results of both, like pristine as well as substituted MOF performance with various temperature, with at different pressure level. So I can able to quantify that MOF will be really going to work for a particular study before it going to synthesize. So it will take, for me, it will take less than a week to complete the whole, generate whole data. So in that point of view, it is very easy. As well as very recently, people are using high throughput screening as well as machine learning application. So in that, what they will do is they will have different connector, different linker. They will connect in different way. And by using these techniques, they will produce more than like 10,000 or 20,000 MOFs, and they will screen the MOF according to their property based on their surface area, pore volume, density, and available pore geometry. So they will cut down the number of MOF from the physical property, physical-chemical property. Finally, they will end up with less than 100 MOF. and they suggest experimentalists to directly synthesize these MOFs, and those experimentally synthesized MOF result as well as theoretically model MOF result both align with each other. So in this point of view, computational chemistry highly helpful to not only for MOF research, it's highly helpful those who are working in material science and those who are really working to solve reaction mechanism in organic chemistry as well as in biology. So in that topic, computational chemistry give its support as well as benefits. And these are the commonly available connector and linker available in the literature to synthesize MOFs. And these are the main applications of MOFs. So due to the presence of this fruitful physiochemical properties of MOFs, by using these crystal structures, we can use these crystal structures for gas storage or gas mixture separation, or in order to want to sense any gas or anything, we can use this as a gas sensor as well as for drug delivery. And some of the MOFs are having open metal site as like active metal site present in the enzyme. So we can use these MOFs for catalytic study, and these MOFs used for proton conduction as well as for water purification. And in this field, widely these gas molecules as well as hydrocarbon materials are tested or used for the application of this material, whether these MOFs, what kind of MOF suitable for example, if you want to capture carbon dioxide, if any MOF which has nitrogen in their linker, or if you have a nitrogen functional group in the linker, that will ready to attract or interact with a CO2 molecule. If we have a CO2 and CH4 in a gas mixture, that will trap particularly carbon dioxide molecule. So in order to, if you want to separate gas molecules, you can use those kind of material. So it's highly depending on what really what we want to do with this material. If you want to separate any C2 hydrocarbons, you can use MOF, different MOFs, because if, for example, if you want to separate ethane and ethylene, by choosing appropriate MOF with appropriate pore structure, you can selectively trap ethane than ethylene, because when we are going to do separation, we need to focus, or we need to keep in mind that what is the kinetic diameter of any gas molecule, what that we going to use. So the kinetic diameter, pore size, as well as temperature, pressure, all these are like interrelated parameters which all determine the property of material to whether this MOF will be good for storage, or this MOF good for particularly separate this mixture. It's all depending on their topology as well as chemical environment present in the MOF crystal structure. We can't say that all the MOFs are very good, but MOF with appropriate pore structure, thermal and water stability, so and those small MOFs we can call as a promising one, and we can use those kind of MOF for any application to store or separate or sense or photochemical studies or whatever it may be. Okay, so what this computational approach differ from the experimental study. So in order to do any experimental things, we need to have the starting material A and B, and we need to keep all the A and B in a conical flask, and we need to put it in a heat bath or a suitable experimental condition to initiate the reaction and to get, in order to get our desired product. And then we will go for the analysis and we'll characterize what's happening. But before going to start the reaction, we know what's going to happen, and whether it will come with our expectation or something is happening new. So in that way, we will do the analysis part. But in the computational point of view, we need to do we need to have like adsorbent and adsorbate material, like material A and B, both need to be optimized before going to do for any calculation. It's like we need to have a pure compound before we going to start the experiment. Likewise, we need to relax the bond distance, bond angle, these parameter, physical parameter we need to optimize. And the crystal structure, most of the in computational study, we get already available crystal structure. We download it from the database and we optimize. If any solvent is there, we just remove the solvent, delete the solvent, and we after that it's just like purifying the starting material for the reactions. Okay, so in order to make interact, we need to have a force field parameter to describe the interaction. So in the force field parameter, which has all the force field parameter, which has bonding parameter, bending parameter, and all the essential parameter. So these force field parameter are transferable as well as flexible. So it should be applicable in all the environment. After that, if we have the parameter, then straight away we start our simulation. In order to characterize MAF for gas storage application, we can use Monte Carlo as well as molecular dynamics. In order to do quantitatively characterize how much amount of gas molecule absorbed in the MAF, we need to do the Monte Carlo study. If you want to calculate the diffusion coefficient, at the time we need to have molecular dynamics study. So this computational work, what I have done is I use multi-scale modeling techniques. That means I'm not in with one software to do the calculation and do the analysis. I use a variety of softwares, and I started to calculate the materials absorption or attraction property from the molecular level to bulk level. So in the output, after the simulation, in the output we can we will get adsorption isotherm as well as diffusion coefficient, and we get this molecular level of information that we can't get it from the experiment. That means you can see the molecule where it's binding, whether it is what is the distance between those molecules, adsorbed molecule from the center of the metal node or metal cluster, or from the linker, what is the bond distance, how it's binding, what is the mode of binding. So all these essential and important parameter, we can get it from the results. And these are the essential equation that we need to describe how this gas molecule should interact with this material. So in most of the thing, we need to have Lennard-Jones potential for the framework atom, as well as for this gas molecule adsorbate. So, and these are the things, and Lorentz-Berthelot mixing rule will help to mix this sigma value as well as epsilon value. So that will give that will make your if you have an accurate collision parameter as well as energy parameter, collision parameter as well as energy parameter, your force field is good enough and more accurate. And if you have the accurate force field, the result what you are getting is exactly matched with the experimental results. And excess absorption, what we get it from theoretical simulation is absolute absorption. So in order from the experiment, we will get excess absorption. In order to compare the theoretical result with the experiment one, we need to convert it. For that, we need a density as well as pore volume of the structure. So that by using this equation, we can just simply convert the absolute absorption into excess. And isosteric heat of absorption will give strength of how the gas molecule interact with the framework. For example, whether it is weakly interact, chemically interact, or physically interact, from the energy value, we can identify how strongly it's binding with the substrate. So far I discussed with you molecular simulation point of view, how the calculations are going. We need force field parameter and blah, blah things. So this graph, you can see, this is obtained from the quantum chemical calculation. So this is simply an optimization graph. The optimization means, as I mentioned, it's more like experimental stuff. So in order to do functionalization or do analysis or further work, first you need to purify your compound. Likewise, here we need to, this is the initial geometry. By using appropriate software, you can build a molecule. But if you can ask me that's the appropriate model. If you ask me, I would say no, that is a crude model. That means you need to refine it. So in order to refine it, we can do optimization first with any molecule. If for example, it's interacting with the gas molecule, and the bond distance, initial bond distance between the metal center to the first atom distance is 2.50 angstrom. You just forget about the bond, it's V ratio. So there is no bond between the metal as well as the gas molecule initially. So when I am submitting the input in the appropriate software, so the initial energy will reduce, and I will get, after 29th or around 30th step, I'm getting this optimized structure. Okay. So what will happening from this energy to this energy? So initially the crude structure has higher energy. And when we are started to do optimization, all this bond length and bond angle and the torsion, everything will relax itself. It's highly depending on the method as well as functional what we are going to use to do the optimization calculation. If you are using a lower level, the calculation will finish within a minute or more. It won't take more than 10 minutes. But really, if you want to have a real structure, which bond parameter and everything, which should match with the experimental available structure, we need, at the time we need to have excuse me, we need to have a very good functional as well as method to do the optimization process. Once we got the optimized structure, normally we do a frequency calculation in order to confirm that it's a minimum structure or not, minimum structure or not. If we don't get any negative frequency, that means that structure is fully relaxed in the potential energy surface. So if it has any negative frequency, that means that the structure is not relaxed fully. So in that scenario, we do a little bit change in the bond distance or angle, and we do resubmit the calculation. Then it will run, and it will take its own number of steps. Finally, it will end up with a fully optimized structure without any negative frequency. So these are the things I really like to share with you before start my work presentation, because I like that once I explain about all these things, it will be easy for you to follow the upcoming slides. So the outline of my presentation is here. So I'm going to give a little bit hydrogen storage. So in this presentation, I'm going to talk about hydrogen storage as well as O2N2 storage and separation. So this is part of my full of my PhD work and part of my current postdoctoral research work. So first, I'm going to give introduction. Okay. So the energy resources are in two types. One is renewable, another one is non-renewable. We highly depending on non-renewable energy resources called fossil fuel, coal, natural gas, and nuclear for our day-to-day life energy purpose. But these energy resources has its own disadvantage or drawbacks, because they are all environment hazardous, because it's producing greenhouse gases and other unwanted side product. And main problem is their prices keep on increasing day by day. And these byproduct coming from these non-renewable energy resources are responsible for acid rains. And nowadays the resources are getting limited. So we are in a shortage of getting this kind of fossil fuels. So at this time, we are in a position to looking or we to get another alternative energy resources or energy carrier to fulfill our requirement. So in this hydrogen is not energy resource. Hydrogen is an energy carrier. When we are breaking HH bond, we will get energy. So this is simple energy cycle or hydrogen economy cycle. When we are electrolysis of water will give hydrogen as well as oxygen. This is fuel cell. So I will come to this part after explaining this. What is happening here when we are in the fuel cell, when we are purging hydrogen as well as oxygen, hydrogen will go anionic oxidation and it will reduce with oxygen in the cathode. The interesting thing and very important thing is this fuel cell, hydrogen-based fuel cell, will produce energy as well as water as a byproduct. So here there is no carbon emission. This cycle sometimes called carbon-free cycle or energy cycles. So this part, so we are producing hydrogen and we are able to produce energy, but we need to store hydrogen. Hydrogen, we all know that it's a diatomic molecule and a highly flammable one. So we need to handle this hydrogen too carefully. So in order to store hydrogen, for example, mobile application or transport application, so we need to have more amount of hydrogen. If you want to replace petrol or diesel, if you really want to, for example, if we want to have a hydrogen-based car or any two-wheeler or anything, we need to have a compact component to store hydrogen molecules. Otherwise, it's too risky and it has its own safety issue. For example, if you want to store carry 1 kilogram of hydrogen in a gaseous form, at least we need a big room size of volume to have a hydrogen molecule, but it's practically difficult. So in the alternative, there are various methods. Hydrogens are used in at present. Those are okay. I will first explain this, then I will go to there. Those are like compressed gas tank or a liquid hydrogen tank. So why we need to store hydrogen? Why we need to have hydrogen for energy application? Yeah. If you are breaking a hydrogen molecule, 1 minute. I'm getting this pop up. Okay. Okay, sorry. So if you are calculating on a weight basis, hydrogen, it has 120 megajoule per kg. It's higher than gasoline. If we are calculating the energy content of hydrogen in the liquid hydrogen, it is 8 megajoule per liter, and the energy content of gasoline is that 32 megajoule per liter. So if for hydrogen storage materials, for if we really want to have transport application, it should come for at least 300 miles or more than 300 miles. In order to satisfy that, we need to have onboard hydrogen storage material should have 5 to 13 kg of H2 molecules. So we really need a specialized material or component to store hydrogen molecules. But in order to to that, we need to come across with all these challenges. One is weight. If we have a more weight, then it will reduce the mileage. And efficiency, durability, refueling time, and finally, the most important one is cost. We need to produce material with a cheaper one. So we need to, before going to get new material, we need to completely understand about the conventional material. So these are the three major ways we can store hydrogen. One is compressed hydrogen tank, another one is a liquid hydrogen tank, another one is a material-based hydrogen storage. So each method has its own advantage as well as disadvantage. In the compressed tank, we need to have a proper cylinder to store hydrogen molecule, and the volumetric capacity also a major thing here. We are getting volumetric capacity of 30 gram per liter, and we need to have pressure of 35 to 70 megapascal pressure. And in the liquid hydrogen forms, we need to liquefy the hydrogen. For that, we need to produce additional energy, and we need to have that setup to keep hydrogen in the liquid form. But compared to the compressed gas cylinder, we are getting a higher amount of capacity when we are using liquid hydrogen. But safety and production cost is the main issue, while you in order to use liquid hydrogen for multiple purpose. And the third and most suitable one is material-based storage. Here, few material I had highlighted. One is metal hydrates, chemical hydrates, and porous materials. So today I'm going to talk mostly about the porous material. So the advantage of using porous material is the guest molecule. That means the gas molecule. First of all, the gas molecule won't chemically bind with the porous material. It will physically bind with the porous material. That means in order to dissolve the gas molecule, we don't need to give any additional energy. We just, by increasing temperature or reducing pressure, it will dissolve the adsorbed gas molecule. And depending on the durability of the material, whether we can use it for repeat cycle or not, it's depending on the material. So in that point of view, porous material are promising one. But in order to get more weight percentage, we can have chemical hydrates, so that there it will break the hydrogen molecule, and hydrogen chemically bind with metal chemical hydrates. But in that scenario, we need to give enormous amount of energy to release the adsorbed hydrogen molecule. So throughout my study, I had used, as I mentioned, I used multi-scale simulation technique. Well, that means I use periodic as well as non-periodic system. That means cluster model and crystal model. So for the non-periodic system, I mostly used Gaussian software to do optimization as well as interaction studies. For that, I used a method called M052X. Here M means Minnesota, because this function was developed in Minnesota by Truller and Groves, and the 05 means the year, and 2X means it has exchange as well as correlation function. So it will help to predict the energy accurately. And the basis set I use is Popul basis set. It has polarization as well as diffusion character, so it will help to predict the it will involve all the essential orbital which will contribute for the interaction. So the basis sets are nothing but they are like mathematical representation of orbital. If you want to use particularly the valency cell, because it will go for the interaction, So we can use that kind of basis set, or if you want to use all the electrons, but that will take more time. It will take more time to finish the calculation, but the once again, the method selection as well as this basis set selection is highly depending on what type of calculation you want to carry out. So if you want to do just optimization, you can use a standard functionals and the basis set, or if you want to look at that, for example, if you want to do some excitation study or proton transfer calculation study or any reaction mechanism study, that time you need to have like specific method and basis set for in order to predict their property. And for the periodic system, I used music as well as RASPA. These two codes are developed by Professor Schnarr, and I use Foresight module, which was a module available in the Material Studio software, which we help to relax the initial crystal structure before do these simulation studies. So the first I would like to talk about hydrogen storage capacity of boranes under aligned cages. So this is a closed-icosahedral borane. We all know that it has 12 vertex and 20 faces. These two, atom number 1 and atom number 12, are called 1 minute. Yeah, these two atoms are called antipodal atoms, and we can able to what I did is I just replaced these two boron atom with carbon, silicon as well as nitrogen. This is a charged cluster. It has -2 charge, and I substituted with the corresponding element, and after that I will show the structure first. Will be more. Yeah. So these are the model clusters. I had model and optimized for in order to study the gas storage hydrogen absorption property of the clusters. So for the clear view, I remove some of the hydrogens here, but the hydrogens are really present during the time of calculation. So in the antipodal position, I introduced mono as well as dicarborane. When we are substituting with the monocarbon, the 2- charge reduced to -1. When I substitute with the 2, the cluster is neutral 1, and with the nitrogen also it's neutral. So yeah, and we measure the distance between the antipodal atom of the pristine as well as heteroatom substituted cluster. There is a drastic change in the cluster, but it's not deform the backbone of the structure totally. There's a deformation, but it's not too much. Yeah, so as I mentioned earlier, it has 20 phases. So we can able to store 20 hydrogen molecules. So these are the hydrogen molecule adsorbed clusters. After the binding, I calculate the binding energy for the each cluster. So it is absorbed from the calculation. The hydrogen molecule, 20 hydrogen molecule adsorbed complex showing higher binding energy. In that, the 2- icosahedral closed borane anion having higher binding energy than the rest of the substituted complex. So this is a binding energy graph. You can clearly see that B12H12 2- cluster having higher binding energy than the substituted one, or the mono substituted having higher binding energy next to 2-, and the neutral cluster having lesser binding energy than the cluster. The one more interesting thing here is all the hydrogen molecule are preferring end-on overlapping. That means head-on overlapping. That means only one hydrogen atom pointed towards the center of the boron gas. That means the cluster itself, it has a 2- charge, and there is a repulsion between the bonded electron of hydrogen and as well as the charge of the cluster. So it prefer to bind head-on overlapping. That means it's not parallel interacting, it's perpendicularly interacting with the center of the molecule. So next, yeah, for the 20 hydrogen molecule absorbed clusters, we calculate what is the binding energy of per hydrogen molecule. So from that also we got among all the clusters, charged cluster having higher binding energy for per hydrogen molecule, but it's still less than the target value. The target value means Department of Energy. They used to fix target for hydrogen storage material or CO2 capturing material capacity. They have their own parameter. If you want to say the material is good for this kind of particular study, that should follow their target value. But according to that target value, the binding energy material, the gas molecule binding energy should be in between 5 to 10 kilocalorie. It should be, that is the pieces of sun range. So if any material falls under that material, we can tell that as a good material for storage purpose. And compared to that, it's very less. But among the cluster, this 2- anion having higher binding energy. So hydrogen. Then I modeled this aluminum cluster. I replaced the boron atom with aluminum and looked at how it behave, whether it is behave like a boron or it's different. So it's also absorbing hydrogen molecule with absorbing 20 number of hydrogen molecule, but the bond distance is high here. The binding energy is also less compared to the boron cluster for the 20 hydrogen molecule. It's here it has here also the 2- ion having binding energy than the rest of the molecule. So here also we calculate the per hydrogen molecule binding energy, but it is less compared to the boron cluster. So this kind of advantage we have with the computational chemistry. See, whether if you want to have a particular group of element in the periodic table, you want to know the characteristics of element and how it will behave before going to synthesis, we can model and we can use the accurate methodology, and we can predict that if it can be synthesized, it will be good or not. We can tell it from the calculation results. Yeah, this one is main one of the interesting thing. So what I did here is I just replaced this existing MOF5 linker with the ticarborane moiety. So it's, as I mentioned, it can able to absorb 20 hydrogen molecule, but when we are incorporating into the into the MOF5 linker modified with this one, this part it has a repulsion, so we can't able to store hydrogen molecule here. But this drum part of the chlorocarborane, we can still have hydrogen molecule, so it's still absorbing 10 hydrogen molecule. But this actual MOF5, it's interacting with it's absorb maximum of 2 hydrogen molecules. So then we calculate the binding energy as well. So the connector parts, it's both modified and pristine one absorbing similar number of hydrogen molecule. But the linker part, as I mentioned, there is advantage of using ticarborane, and you can get more number of hydrogen molecule in the linker. So that will also help to improve the material quality as well as property towards the application. So one main interesting thing is, so we had published this work 2012, yes, 2012. So after 2012, these two structures experimentally synthesized, and they proved that the presence of it can be, first thing is we can able to synthesize chlorocarborane based metal organic frameworks with various topology. That's the first point. The second one is this material also showed that this material absorbing hydrogen molecule with high amount than the pristine one, like benzene one, benzene substitute pristine cluster. So there is a hope that if we can model properly, model any system, that can be easily synthesizable. So these are the evidence for that. And the second, I calculate the hydrogen storage property of carbon-based material, and I decorated those carbon material with alkali and alkaline earth metal cations. So in the previous work, I used anionic cluster. So here I use cationic cluster. So I want to know how the anionic and how the cationic cluster behaving towards hydrogen adsorption. But the same the way I choose boron and the way I choose carbon, because they are like low weight material, it won't interrupt when we are coming to calculate the weight percentage of molecule. So they are like low molecular weight of cluster, so it won't affect the weight percentage of the system. So for that, I have chosen cubane, adamantane, and cyclohexane. They are like a cage material available in, and this material I introduced the metal cation over the substrate in this manner. Okay, so what are the metal ion I had chosen? So I selected S block elements, lithium, sodium, potassium, magnesium, and calcium. These metal cations are widely used to study hydrogen storage application purpose. So initially, first what I did is I just optimized the cubane itself. Then I introduced metal cation over the cubane. I placed the metal ion face of the cubane, and in between this carbon-carbon bond over here. So first I figure it out where the metal ion tightly bind with the substrate. So then I put hydrogen molecule one by one. From the optimization results, I found that lithium cation can absorb maximum of 4 hydrogen molecule, and sodium is absorbing 5, and potassium absorbing 9. So when we are moving from lithium to potassium, the ionic area of the material is increased, the van der Waals area of metal atom is increased. So if that means we are getting more room to accommodate hydrogen molecule. But the one disadvantage is the distance between the metal and hydrogen will increase. That means if we are really going to do for a real study, in that point of view, we can't use potassium, but in that case, we can use lithium or sodium. The distance are very less; it's less than 2.5 angstrom. So that means it's strongly bind, but it's not chemically bind. And one of the thing what we need to know is when we are having this anionic cluster, that time hydrogen molecule form perpendicular type of interaction. That means end-on interaction. But here the metal atom, hydrogen is forming T-shaped complex. That means both the hydrogens are trying to interact with the metal cation. And for the magnesium and calcium, the same scenario. When we are moving from magnesium to calcium, the van der Waals density of the metal atom increase, so we can store more number of gas molecule over the metal atom. After the optimization, I calculate the binding energy. So the binding energy complex are quite interesting and they are high. So compared with lithium and magnesium complex, magnesium is showing higher binding energy, and among the three, lithium is showing higher binding energy for per hydrogen molecule, and the calculated weight percentage also higher than the target value. So it's 6.8, here it is 7.3. The target value is around 5. So this material fulfill the target value, but in order to do the practical application, still we need to work on this material more. So we figure it out why this calculate the bond distance of the metal atom before and after hydrogen binding. the difference is not that much, it's little bit. And we calculate the charge of the metal ion before and after hydrogen binding. There is a charge transfer taking place. You can see the metal charge is 0.948 in the before hydrogen. After hydrogen binding, it is 0.618. Actually, this metal environment, what it's doing is it's just polarizing the absorbed hydrogen molecule, but it's not totally polarizing. It's a little bit polarization. So that little bit change always make chemists interest. So that little bit charge transfer trapping hydrogen molecule with required per hydrogen molecule binding energy, as well as the substrate low molecular weight substrate, helps to increase the gravimetric density of the complex. So similarly, we done with cyclohexane and we calculate the binding energy. Here also we found that magnesium tetrated complex showing higher binding energy. There is a charge transfer taking place between the metal atom and the hydrogen molecule. And this one is for adamantane. So here also we got similar kind of result. Among the three substrate, magnesium doped complex showing higher binding energy than the other four metal cation. So what we did is we just took this metal organic polyhedra. MOP means metal organic polyhedra. They are just different than metal organic frameworks. They are discrete crystal units. It's like a ball-like structure. It don't have a extended crystal structure. It's like a separate one. So what I did is I just replaced the existing benzene group with the cubane molecule and calculate the basic fundamental unit. These are the basic fundamental units available in the crystal structure. So we quantify quantum chemically what is the hydrogen storage capacity of the material, and we found that the presence of cubane linker helps to increase the number of hydrogen molecule on the linker. So if we can able to synthesize this kind of model, this will highly helpful to improve the hydrogen storage property of the material. Yeah, so compared to anionic as well as cationic cluster, we found that cationic clusters are good enough to store hydrogen, trapped hydrogen molecule with appropriate with required binding energy, and if the substrate is low molecular weight, that will help to improve the weight percentage. So in this one, what I did is I had chosen polyhydroxy adamantane and introduce open metal site on the polyhydroxy adamantane unit and calculated their hydrogen storage property. So for that, I had used. So before going to talk about my work, I just briefly explain how this open metal site help to improve the binding energy. These are the theoretical model one. Here, catechol kind of moiety functionalized over the metal organic framework MOF5, and they calculate the binding energy, I mean weight percentage of the material near room temperature. This material presence of open metal site help to improve the material weight percentage around like 3.4 weight percentage near room temperature. And this one is called porous aromatic framework. This one also a novel porous material which don't have any metal. It's like actually it's like a diamond-like structure. If you are imagine if carbon is here, that carbon was replaced with the benzene moiety. So you can extend the number of benzene in the linkers. By doing that, you can improve the surface area, or you can tune the pore volume. So that may help to specifically store or separate particular gas molecule. So here they introduced a double catechol kind of open metal framework over the single linker, and they also proved that the presence of magnesium and calcium open metal site help to improve the weight percentage of material at near room temperature with a 100 bar pressure. So in my PhD group, my senior also introduce this kind of various transition metal cation in the linker of IR of 9, and he calculated the presence of metal transition metal. Phosphor transition metal helps to improve the per hydrogen molecule binding energy. These are quantum chemical results. Yeah. So here what it is, as I mentioned earlier, the magnesium and calcium are acting well than lithium and sodium. So in this work, I had chosen magnesium and calcium. So as I mentioned, this is the polyhydroxy adamantane. So the beauty of this adamantane is, adamantane itself, it's like a three-dimensional structure. It has 4 CH and 8 CS2 group. Yeah, 4 here. So we can able to introduce more number of hydroxyl group. That means we can have a polyhydroxy adamantane. There are experimentally, these structures are experimentally available. So I had chosen this structure, and we can able to model OH group with in a various position. So here you can see this is a single metal ion anchored polyhydroxy adamantane, and this one is two metal anchored complex. For the two metal anchored complex, I have model 1, 2, 3, 4, 5. Yeah, for 3 metal 6, and for 3 metal we have 2, and the maximum of over the single adamantane complex we can introduce like 4 open metal site, and there is no formation chance for the formation of metal cluster formation, because the bond distance between the two metal is more than 7 angstrom. So there is no chance for the formation of metal cluster formation. So similarly, we introduce calcium. So for calcium also, we can able to, though the binding area of the calcium is higher than the magnesium, we can have still 4 calcium on the moiety. So after incorporated with the metal atom, then I moved to study the hydrogen storage property. So I started from 1 hydrogen molecule. So magnesium can able to absorb maximum of 4 hydrogen molecule without any hydrogen-hydrogen repulsion, and the bond distance also less than the, it's not located far away from the center, and maximum of 4 metal atom adsorb complex adsorb maximum of 16 hydrogen molecules we can store over single polyhydroxy adamantane unit. Yeah, and I calculate the binding energy as well. So for the 4 hydrogen molecule, the binding energy per hydrogen molecule is close to the 5 to 10 kilocalorie range, and for this one 16 hydrogen molecule having higher binding energy as well as higher weight percentage. And this is for calcium system. Calcium we can store maximum of 16 hydrogen molecule. Yeah, see. So we can, for calcium, sorry, we can store 24 hydrogen molecules. For single unit, we can store 6 hydrogen molecules. Still, there is no repulsion between hydrogen molecules. And we calculate the binding energy. So here also we got that maximum number of hydrogen molecule absorbed complex having higher binding energy as well as weight percentage. So that means we can have this moiety alone, or we can have a functionalization of this adamantane in appropriate metal organic framework, and further we do this open metal site on that, and we can use that for particular hydrogen storage application purpose. Yeah, so after got this value desired per hydrogen molecule binding energy as well as gravimetric density. We theoretically want to know whether this material will be stable at near room temperature or not. So in order to do that, I had done ADMP study. That means atom density matrix probation technique is available in Gaussian. So I had chosen that method by using this M062X. This also Minnesota functional, was produced in the year 2006. And I had carried out the simulation with 50, 100, 200, and 300 Kelvin with a time frame of 1 picosecond. And the time interval between the steps are like in between 1 femtosecond. So in order to maintain the temperature, I had used velocity scaling thermostat that will help to keep the temperature into the desired temperature what we want to run the calculation. So these are the snapshots I'm sharing. So at 15 Kelvin, there is no desorption taking place, as well as 100, as well as 200. So up to 200 Kelvin, the molecule is not ready to release hydrogen molecule. But after at 300 Kelvin, 1 picosecond, the moiety is dissolving 2 hydrogen molecule. If you are increasing time, obviously the molecule will release more hydrogen molecule. The one important thing is the structure of backbone of the moiety is not deformed or ruptured near room temperature. That means it's forming a stable substrate. So this means that we can use this material for to trap hydrogen molecule near room temperature. And this is for magnesium system with various time steps. So it started to lose hydrogen molecule from 400 femtosecond. And these are the subsequent snapshots. And this is for calcium. So as I mentioned earlier, the radius of the metal and the distance between the absorbed hydrogen molecule metals also matters. That's also determine whether the hydrogen molecule will tightly bind with the desired binding energy or not. So here up to 100 Kelvin, there is no desorption taking place. But near 200 Kelvin, there is only one hydrogen molecule dissolved from the moiety. But at 300 Kelvin, more number of hydrogen molecules started to dissolve. So in order to, what we observe from this study is metal compared to anion, metal cation with appropriate substrate, and possible we can create more number of metal cation on the single substrate will help to improve the binding energy of the material. So these are all like multi-scale modeling techniques. That means now I calculate the binding adsorption, mode of adsorption, bond distance, as well as number of hydrogen molecule absorbing over the metal over the single unit. So from which I will model bulk system, then I will go for the bulk storage study. So that means I have a data for molecular levels as well as bulk level. So this kind of multi-scale modeling technique will give you a complete picture about fundamental as well as quantum level as well as molecular level, bulk level, adsorption or molecular property of the material. And these are the snapshots. So here it started to release hydrogen molecule from 500 femtosecond. So in the subsequent femtosecond, it's losing more number of hydrogen molecules from the moiety. So at 900 femtosecond, you can see clearly it's far from the moiety. So it's completely dissolving a molecular hydrogen. Yeah, so in the so far presentation, I highlighted the importance of cluster model approach. That means in the quantum level, I calculate the gas storage property. But here, actually, I model a real crystal structure with appropriate linker. For that, I had chosen these carbon flakes. So these are the linkers I had chosen, and I use MOF5 to build this new linker-based material. So I had also concentrated on the shape of the linker, whether it may play a role or not. So I want to know that. So I had chosen circular, rectangle, and rhombus linker-based morph structure. So these are the unit cell as well as supercell of the circular linker-based morph, and this one is for a rectangular linker-based morph, and this one is rhombus-based morph. So you can see from the structure, there is a chance, there is a different kind of pore structure exists in these crystal units. So the yellow color sphere, what it's highlighted that it has this much amount of void space inside the crystal structure. So this will help you to store more number of hydrogen molecule when we are changing the pressure or temperature. I will tell those things in the upcoming slides. So we have a crystal structure, and we need to know how the pore structure or pore size distributed over the crystal structure. What kind of pore, whether it has a single pore or it has a di pore or it has a multiple pore size in a single crystal. So from the pore size distribution analysis curve, we got that this model cluster cubic kind of IR morph having two different pores on it. Yeah, and so the physical structural property of the morphs, we compared the model morph surface area, density, pore volume with the already reported value. So from the result, we found that so we replaced actually this IR morph 1 structure. Oh, sorry. With the we are in the model this IR morph H1, H2, and H3. So the introduction of these carbon flakes will highly helpful to improve the surface area, density, as well as will help to improve the pore volume. Yeah, so this kind of approach helped to improve the physical parameters of morphs. Then we moved into quantify how this molecule, this model morph will whether it will absorb a gas molecule with bulk amount, or it has any drawback or not. So in order to do that, we need to have the Lennard-Jones potential like sigma value as well as epsilon value. So here I have used methane, hydrogen, as well as carbon dioxide. Here, excuse me. Methane and hydrogen are treated as a united atom approach. The one advantage here is if you are using more number of atom in computational study, your calculation running time will increase. So in order to reduce, as well as we should not compromise in the accuracy. So for that, we already have a force field parameter for the gas molecule, what I had used. So here we use the united atom approach. That means the carbon methane molecule is considered as a round. It's like a sphere. It's like a single sphere model. What you call it, say. But carbon dioxide is treated as a three-set model. It has carbon as well as 2 oxygen in the molecule file, and which has their own charges as well. So, but in the united atom, we normally won't go for charges. And for the framework atom also, we need to have a partial atomic charge in order to mimic the experimental condition. That we actually need a real system, so we need to give all these parameter carefully and correctly to the in the input file. So these are the simulation snapshots absorbed from various pressure range. See, here you can see the advantage of using this linker. I will tell you here. So if we have a single benzene molecule in the connector part, we can able to store 2 hydrogen molecule at very low pressure, but maximum of at higher pressure we may have more number of molecule. But if you have more number of attraction site in a linker, that will help you to improve the material gas uptake property at moderate as well as higher pressure. So here at very low pressure, the gas molecules are trying to populate over the most active site is metal cluster site. So the gas molecules are 1 minute. Yeah. Here you can see the gas molecules are populated over the metal cluster unit, connector unit. At moderate pressure, the gas molecules are trying to populate over next to the metal cluster. It's trying to populating over the linker atom. That means if your material point, the gas molecule is find more attractive place. Next, when we are increasing pressure, it will find more suitable adsorption site. If we can able to model material in that point of view, so we can trap more number of gas molecule over the linker. At higher pressure, it will highly populated in the pore volume. So that's the thing. So your material, if your MOF having attractive site, required linker with moderate or bulk pore volume, so you can use this material to store or separate any gas molecule with various. So you can adopt this material in various pressure range as well as temperature. So all these simulation run at 77 Kelvin, and this one at 298 Kelvin. So here you can see when we are at room temperature, the material is not absorbing more number of hydrogen molecules. That means it doesn't have any open metal site or effective binding site. So we can use this material. This material can't accept store hydrogen molecule near room temperature. That means we can use this material for hydrogen purification. If something is attracted towards by bonded this linker or waste binding with this metal cluster, if you have hydrogen and those gas, that in that scenario this material can be used for hydrogen separation. So hydrogen molecule won't bind with that much effectively with this material, so we can use this material for separation. And these are the weight percentage similar isotherm curve. So normally isotherm curve we will draw in between amount of absorption versus pressure. So here I draw with the weight percentage in the x-axis, y-axis, and the pressure in the x-axis. So here you can see. So among the three model MOF, this IR MOF H3, that means the MOF linker with Rampus mighty showing a higher weight percentage at 77 Kelvin, and the gravimetric density as well as the volumetric density also for those MOF is higher than the circular and square linker modified MOF. So what I aimed, what my objective is, whether the shape of the linker play a major role or not. So from this I can understand that the shape of the linker also play a major role because it's highly influence the crystal structure as well as pore volume of the MOF. So that's the thing. And this is for at 298 Kelvin, that is for 77 Kelvin. So here at 298 Kelvin, the MOF, none of the MOF material attaining the saturation points, still the absorption curve is a straight line. So a lot of space, still we need to have a lot of room to store hydrogen molecule, but near room temperature, the molecule is not, material is not absorbing more number of molecule. And this is for methane. So here also the same scenario. The methane molecule try to populate initially over the connector atom, then it's populating over the wall of the linker surface. Then the pore volume of the material is occupied with the gas molecules. So here also we did the I did the simulation at 298 300 Kelvin. So still it's not attain the saturation point. Still a lot of room is there for to accommodate the gas molecule. I run the simulation for up to 100 bar. So there is no much absorption of methane, and this is for CO2. So we run the simulation for 50 bar, and we got here IR MOF H1 showing higher uptake for uptake than the other two MOF. So we can use this IR MOF H1 for CO2 capture as well as hydrogen purification. So that's what we got from this simulation results. Yeah, so now in the final presentation, I'm going to talk about my current postdoctoral research work. Here I have used heterometallic metal organic framework for air separation. The air separation, it has nitrogen and oxygen, so we need to what we really focused in this work is we want to trap oxygen molecule instead of nitrogen molecule inside inside the MOF. Conventionally, if you are using zeolite or MOF with mono metallic metal organic MOF with monometallic moiety, which are attracting towards nitrogen molecule and it's releasing oxygen molecule, but we want to trap oxygen molecule in order to for the purification as well as for a variety of industrial application, because oxygen has its own industrial application, various industrial applications. So in order to do that, we have oxygen with high purity. So in that scenario, we need to trap oxygen molecule. So for that, I had used PCN 250. Here, PCN means it's porous coordination network. 250 is in that series. It's the 250th MOF, synthesized MOF. So this is the PCN 250 crystal structure. Excuse me. Yeah, and this is the connector part. Here it has a triaxo metallic center which connected with azo linker, and it has OH group, one OH group in the metal surface which is chemically bonded with the iron atom. The pristine PCN 250 cluster connector unit has 3 metal center, and the 3 metals are Fe3 oxidation state, and they are in high spin state. So what is the beauty here is when we can able to, when we are removing this OH moiety, experimentally it was proved that when we are removing this OH moiety, what it's doing is it's reducing this Fe3 ion center into Fe2 center. So there is a heterogeneity begin when we are removing this oxygen OH group. And one more interesting thing is if it is forming a heterometallic center, we can able to substitute or replace 2+ metal with various metal cation with 2+ oxidation state. There is a chance. So there are experimental report available with various divalent metal substituted, transition metal substituted, these PCN 250 clusters are there. But in this work, what we did is we replaced this Fe2+, this OH, and replaced with chromium, manganese, and cobalt, nickel, as well as zinc 2+. So we created various this heterometal substituted moiety. So here you can see spin density picture of the corresponding cluster models, what we model with various first row transition metal metal group. Yeah, the reason why I have shared this picture is before going to do grand canonical Monte Carlo simulation to get the isotherm graph, we want to know what it's happening when we are replacing with various different metal atoms. When we are changing the metal atom, the charge, spin density changes. So whether it will play a major role to trap interaction of, whether it's playing any key role to find the interaction of gas molecule energy. So we want to know that. We got the spin density graph from quantum chemical calculation. And before going to calculate the connector part, storage property, we calculate the linker part, adsorption strength of oxygen molecule as well as nitrogen molecule. And from the study, we found that there is oxygen molecules are binding a little bit stronger than nitrogen molecule over the linker. Yeah. So this is the complete picture of how the gas molecules are binding over the oxygen and nitrogen molecule binding over the model clusters. So this is for the pristine system, which has all the metal with +3 oxygen state. Here it has +2 chromium with +2 state and the other two metal in +3 state. So we introduce the hydrogen, oxygen as well as nitrogen molecule over the 2+ as well as 3+ oxygen state, and we found that the gas molecule effectively binding over the 2+ state than the 3+ state. This may be due to the amount of charge transfer happening from the +2 metal to the oxygen atom, from the +3 metal oxygen atom. So it is happening quite effectively from chromium to 2+2 oxygen atom. And the corresponding calculated binding energy graph also shows that compared with the nitrogen, oxygen is binding effectively over the model cluster, and the corresponding relative binding energy graph also shows that oxygen is selectively binding over the cluster than the nitrogen molecule. And to qualitatively, in order to validate our quantum mechanical result, we done GCMC simulation studies for all these 7 clusters. So we had chosen a supercell and calculated their storage property at 298 as well as 273 Kelvin with the pressure range from 0.1 bar to 1 bar. So it's all like real room temperature condition. We found that chromium substituted complex absorbing oxygen molecule effectively than the nitrogen molecule at 298 as well as 273 Kelvin. And the simulation snapshots also shows that the nitrogen molecules are always try to populate over the linker atom, and oxygen molecule try to populate over the open metal site. So from this study, we found that we can, if you have heterometallic moiety, that will highly helpful to selectively store oxygen than nitrogen. So this kind of model study will highly helpful to people who are working in this area, or this kind of information highly helpful to the experimentalists to selectively synthesize that particular material for that particular application purpose. Thank you for your kind attention.
Thank you, Dr. Gopal Swami, for nice presentation. Is there any questions from audience? Actually, we have 2 questions.
Yeah.
Sir, one of our participants has asked this question. Most of the metal you used for calculation is oxophilic. So can you comment on the oxophilic character of the metal you used with interaction of hydrogen molecule?
Yeah, we are creating the open metal site that will highly helpful to polarize the absorbing hydrogen molecule, even though it's oxophilic. So this metal cation will help to improve the binding energy. So we are looking into in that perspective. So we have chosen all these S block as well as first row transition metal.
Okay, sir. The next question is, can you comment on the most suitable metal for hydrogen storage from the metals which you have came across?
We can't generalize, but we can pick at least one or two metal from the corresponding element group. Like from the S block element, we can use lithium as well as magnesium. In the transition metal, depending on the chemical environment, we can use for hydrogen storage, we can use scandium is there, we can use scandium as well as titanium. But in that scenario, if we are going to transition metal, we should compromise. We supposed to compromise with the weight percentage, as well as we need to give external energy, because hydrogen molecule effectively binding with this transition metal. So we need to give additional amount of energy for the desorption studies, and there is a chance for deformation of structure when we are using highly reactive this transition metal. But I would say S block elements are more appropriate and suitable for hydrogen storage. It's all depending on the chemical environment, that in which environment the metal is present. That also we need to look at in for this kind of particular application purpose.
Thank you, sir. These are the questions in the chat box.
Oh, thank you.
Thank you.
Thank you, Dr. Gopalswamy, for a nice presentation. I would like to thank you for you and the other participants on behalf of our Department of Chemistry and FCS 2020 team. Thank you all.
Thank you so
tomorrow,
much for the invitation. It's a pleasure for me. It's my great honor that I present my work in FCS 2020. I would like to thank and convey my thanks to the organizer, as well as Department of Chemistry, Bardiyar University, as well as all the audience and all the co-presenters. I would like to thank everyone. So I take this moment to thank everyone. Thank you all.
Thank you all.
Okay, all the session of today is ending. All the session is ending now for today, and tomorrow all the participants have to come around 9:45. The sessions 6 will be around 10 a.m. tomorrow. Thank you.